Imagine a world without antibiotics. It's a world we can barely fathom today. A world where a simple scratch could be deadly, where childbirth, a moment of joy, was often overshadowed by the fear of infection. Before the discovery of antibiotics, these were the harsh realities of life. Infectious diseases ran rampant. Tuberculosis, pneumonia, and sepsis were constant threats. A minor wound could quickly turn fatal. The discovery of antibiotics revolutionized medicine, marking a turning point in human history. It all began with a touch of serendipity and the keen observation of a brilliant scientist. In 1928, Scottish scientist Alexander Fleming made a groundbreaking discovery that would change the course of medical history forever. Returning from a holiday, he noticed something peculiar in his laboratory that would soon capture the world's attention. A petri dish containing bacteria had been accidentally contaminated with mold, an event that seemed trivial at first glance. But what really caught his eye was a clear zone around the mold, where the bacteria had stopped growing, indicating something extraordinary was happening. This mold, Fleming realized, was producing a substance that could kill bacteria, a discovery that was both unexpected and revolutionary. He named it penicillin, after the mold species Penicillium notatum, marking the birth of the first true antibiotic. This accidental discovery marked the beginning of the antibiotic era, a period that would save countless lives and transform medical practices. Fleming's findings opened up a new frontier in medicine, leading to the development of treatments for previously incurable diseases. Scientists realized that nature held the key to combating bacterial infections, inspiring further research into natural sources of antibiotics and other medicines. Antibiotics are powerful weapons against bacterial infections. But where do these wonder drugs come from? The answer lies in the microscopic world of bacteria and fungi. These tiny organisms, often seen as harmful, are the original producers of many antibiotics. In the constant battle for survival, microorganisms have evolved to produce substances that give them an edge over their competitors. These substances, known as antibiotics, inhibit the growth of other microbes, allowing the producing organism to thrive. By harnessing these natural defenses, scientists have been able to develop life-saving medications. Discovering a new antibiotic is no easy feat. It's like searching for a needle in a haystack. Scientists screen thousands, even millions of microorganisms to find those with potential antibiotic activity. The process begins by collecting soil samples, water samples, and even samples from extreme environments like deep-sea vents. These samples are teeming with diverse microorganisms, some of which may harbor the secrets to new antibiotics. Once collected, these samples are carefully cultured in the laboratory, and the microorganisms are screened for their ability to inhibit the growth of harmful bacteria. Section 5. Growing the good guys. Cultivating antibiotics. Once a promising microorganism is identified, the next step is to grow it on a large scale. This is done in large, sterile vessels called fermenters, where the microorganisms are provided with the optimal conditions for growth and antibiotic production. The process of fermentation is like brewing beer on a massive scale. The microorganisms multiply rapidly, producing the desired antibiotic as a byproduct of their metabolism. The fermentation process is carefully monitored to ensure optimal antibiotic production. Section 6. From broth to pill extracting and purifying antibiotics. After fermentation, the antibiotic must be extracted and purified from the fermentation broth. This involves a series of steps, including filtration, centrifugation, and chromatography. Filtration removes any solid particles, while centrifugation separates the liquid broth from the microbial cells. Chromatography techniques are then used to isolate and purify the antibiotic from the remaining mixture. The purity of the antibiotic is crucial to ensure its safety and effectiveness. Section 7. Ensuring safety and effectiveness. Testing antibiotics. Before an antibiotic can be made available to the public, it undergoes rigorous testing to ensure its safety and effectiveness. This involves preclinical testing in the laboratory, followed by clinical trials in humans. Preclinical testing evaluates the antibiotic's activity against different bacterial strains, its potential toxicity, and its pharmacokinetic properties, how the drug is absorbed, distributed, metabolized, and eliminated by the body. Clinical trials involve testing the antibiotic in human volunteers to assess its safety, efficacy, and optimal dosage. Only after passing all these tests can an antibiotic be approved for use in humans. Section 8. Beyond nature's pharmacy, synthetic antibiotics. While many antibiotics are derived from natural sources, scientists have also developed synthetic antibiotics. These are created in the laboratory, often by modifying the structure of existing antibiotics to enhance their properties or overcome resistance mechanisms. Synthetic antibiotics offer several advantages. They can be produced more efficiently and cost-effectively than their naturally derived counterparts. They can also be designed to target specific bacteria, reducing the risk of disrupting the body's natural microbial balance. The development of synthetic antibiotics is a testament to human ingenuity in the fight against infectious diseases. Section 9. The ever-evolving challenge: antibiotic resistance. The discovery of antibiotics revolutionized medicine, providing a powerful tool to combat bacterial infections that once claimed countless lives. However, the fight against these infections is far from over. Bacteria, with their remarkable ability to adapt and evolve, have developed resistance to many antibiotics. This adaptability allows them to survive even in the presence of drugs designed to kill them. Antibiotic resistance is a major public health threat, posing significant challenges to healthcare systems worldwide. It can lead to longer hospital stays, higher medical costs, and increased mortality. It occurs when bacteria change in ways that reduce or eliminate the effectiveness of antibiotics designed to kill them. These changes can be subtle yet powerful, rendering treatments ineffective. This can happen through various mechanisms, such as mutations that alter the bacterial target site of the antibiotic, making it harder for the drug to bind and do its job. Another mechanism is the acquisition of genes that encode for enzymes that inactivate the antibiotic, breaking it down before it can take effect. Overuse and misuse of antibiotics are major contributors to the development and spread of antibiotic resistance. This includes not completing prescribed courses, using antibiotics for viral infections, and the widespread use of antibiotics in agriculture. Public education and responsible antibiotic use are crucial in combating this issue. By understanding the importance of proper antibiotic use, we can help preserve the effectiveness of these vital drugs for future generations. Section 10. The future of antibiotics. A call for innovation. In this section, we delve into the critical need for innovative solutions in the realm of antibiotics. The journey of antibiotics, from their serendipitous discovery by Alexander Fleming to the development of synthetic versions, has been marked by remarkable progress. These life-saving drugs have revolutionized medicine, turning once deadly infections into manageable conditions. However, the emergence of antibiotic resistance poses a significant threat to global health. Bacteria are evolving, becoming resistant to existing treatments and making infections harder to treat. This resistance can lead to longer hospital stays, higher medical costs, and increased mortality. To stay ahead of this evolving challenge, continued innovation in antibiotic discovery and development is crucial. Researchers are tirelessly working to find new antibiotics and alternative treatments to combat resistant bacteria. Scientists are exploring new avenues, such as phage therapy, which utilizes viruses that infect and kill bacteria. This method offers a targeted approach, potentially reducing the risk of resistance. Additionally, antimicrobial peptides, which are part of the innate immune system of many organisms, are being studied for their potential to fight infections. These peptides can target a broad range of pathogens, offering another line of defense. Public awareness campaigns are also essential to promote responsible antibiotic use and reduce the spread of resistance. Educating the public on the dangers of misuse and overuse of antibiotics can help preserve their effectiveness for future generations. The future of antibiotics depends on a multifaceted approach, combining scientific advancement, public education, and responsible stewardship of these life-saving medications. Collaboration among scientists, healthcare professionals, and the public is key to ensuring that antibiotics remain a powerful tool in our medical arsenal.